We are used to seeing DNA represented as a rather neat sequence of letters arranged along a double helix. Straightened out, the human genome would be about 2 meters long. Every cell contains a full copy of the human genome. But a cell is 1/100th the size of a grain of sand, so the DNA has to be coiled, folded, squashed and squeezed to fit inside. You might think it ends up like the very worst sock drawer, just a tangle of material. And in fact, this is how it looks inside the cell. But there is a logic to the apparent chaotic arrangement, just as you might pack a travel case, with the things you'll need placed where you can reach them. DNA strands fold themselves into different 3D configurations, so that sometimes certain sections will be hidden, but become accessible when needed, so that the cell can use the right instructions. 98% of the DNA in our body is non-coding. That means it does not carry the information needed to build proteins. Non-coding was called junk in the past. Today, we know this is far from the truth. Scattered throughout non-coding DNA is a plethora of so-called regulatory elements, including enhancers, silencers, and insulators. Babraham Institute researchers have shown that the location of a gene inside a cell, as well as its position in space relative to other genes, can drastically change how a gene behaves. Our aim is to find these regulatory elements and to understand which genes they control. We study these questions using pluripotent stem cells, cells that have the potential to create all cell types in the adult body. Because they contain all the information to make a complete adult human, these cells can replicate and develop into the different parts of the body. The regulatory elements function like molecular switches to control which genes are active and thus produce proteins in which cells. Just as you would pack your case differently according to the needs of your journey, so the arrangement of the DNA inside the cell varies according to its destination. It's as though you repacked your suitcase at each stage of your journey. This process of gene expression control is vital to allow cells, which all contain the same genes, to specialize in order to carry out different tasks and to help them respond to changes. A breakdown of gene expression control may be a cause of disease as cells are misdirected or DNA is poorly arranged to perform the functions for which it is intended. By uncovering the molecular mechanisms by which regulatory elements interact with their target genes in the three-dimensional space of the cell nucleus, we aim to understand how altering the function of regulatory elements can lead to developmental malformations and disease. Dr. Stefan Schoenfelder's research into this area has led to many advances in the field. He has helped set up a company called Enhanced 3D Genomics, which works to bring this new technology into wider use to better understand the links between gene enhancers to their target genes and unlock their potential for new healthcare therapies. The Babraham Institute undertakes world-leading research into understanding the biology of how our bodies work, including what changes as we age and during disease. You can discover more about the breakthrough work of the Schoenfelder Lab on the Babraham Institute website. The Babraham Institute. Share the discoveries. Find out more online.